So this set of notes is about PCR. So polymerase, and you guys know this, PCR stands for polymerase chain reaction. I put this little picture on here. Okay, so it kind of summarizes the steps. So we're gonna denature, we're gonna anneal, and we're gonna extend. So we'll start at the beginning and go through all of this. So PCR is polymerase chain reaction, and it's used to make many copies of DNA. So it was discovered by Kary Mullis in 1984, and he got the Nobel Prize for it in chemistry in 1993. So if you Google it, you'll find some stuff. There were patent issues and lawsuits and stuff about who owns the patent and who owns the intellectual property rights to it. So that's pretty normal for a lot of science stuff. So basically what PCR does is what we're going to do is we're going to amplify a specific target sequence of DNA. So we're going to look at a specific region of DNA and make more copies of it. So the things that are used to make the specific pieces are primers. Okay, so primers are designed to target the piece. They're specific to the piece of DNA you want to amplify. There is such a way to do random replication, and there are some really short primers that you buy, and you can just randomly replicate pieces of DNA and then try to put them together. So we can actually, using PCR, make pieces up to 6 kilobases big, so 6,000 base pairs long. So there is a limit. You can't just make huge pieces of DNA. And so this picture is Kary Mullis down here. Okay, so here is the overall summary of PCR. So usually you do about between 30 and 40 cycles, and there are 3 main steps that are considered a cycle. So the first step is denaturing. Okay, so in denaturing, what we're going to do is we're going to take the double-stranded template DNA, and we're going to break it apart. We want to make it single-stranded. So when we break it apart, we heat it up to a high temperature. This says 94, lots of times it's 95 degrees Celsius. And it can be anywhere from 1 minute to an initial denaturation of 5 minutes. Okay, step 2 is annealing. So annealing is when the primers are going to bind to the broken apart double-stranded DNA. So the primers will bind, okay, or stick, so anneal. And annealing temperature is going to vary. Okay, so it can be between 40 and 60 degrees. Okay, and then the next step is extension. So extension is where we're going to use an enzyme. We're going to use a DNA polymerase. And we're going to add nucleotides. And we're going to end up, it works at 72 degrees Celsius. And the time for extension can vary. Usually it's about 2 minutes or a minute. Okay, so denature, anneal, extend. Okay, so whenever you go to do a PCR reaction, there are certain chemicals that you have to have in order to make it work. So the first that I have listed on here is template DNA. So this is the DNA that you're going to try to amplify. So DNA you want to amplify. Okay, then you have primers. Okay, so the primers, you're going to have one that goes one direction and one that goes the other direction so that you can amplify a specific piece of DNA. You have to have a DNA polymerase. Okay, so that's an enzyme that's going to make DNA. The one that we use is called Taq polymerase. And Taq stands for Thermus aquaticus, so T-A-Q. So Thermus aquaticus was found in the hot springs in Yellowstone. So it's a DNA polymerase that can actually withstand high temperatures. so it can go through the cycling process of PCR and not break down very quickly. Okay, the fourth thing that you have to have, and they say DNTPs. So DNTPs are the A's, the T's, the C's, and the G's. So these are the nucleotides. This is what you're going to use to make DNA. Okay, then we also have magnesium chloride, and that should be a little too. Okay, so magnesium chloride is necessary. You need the magnesium in order for the enzymes to function. Okay, then you're going to have a buffer, so it's going to help maintain the pH. And then you're going to have water. Okay, so all of those things have to be in the reaction. Okay, nowadays, when we go to do a reaction, lots of times we buy all of these things. Okay, so the polymerase, the A's, T's, C's, and G's, the magnesium, and the buffer. I guess I should put it down here. These are in what we call a master mix. So we buy them and they're already mixed together. They're ready to go. Lots of times they have a color in them. So the one we used in lab last week actually was tinted blue so that you know when you add a master mix. Sometimes the one we use is green. Okay, so we used to buy them separately. Now we buy them all together. Okay, so now the reaction mix, they're put into these little tiny thin-walled tubes. So the tubes are thin-walled, so we can change the temperature easily. So when we're cycling from 94, 95 or 94 to 60 to 72, we want to be able to change the temperature of the liquid in here quickly. So the tubes are really small. The walls of them are really thin. And that's so that we can heat it and cool them faster. Okay, the reactions are cycled. I don't know that the tubes are cycled. I would say the reactions are cycled in a thermocycler. Okay, so this is what ours looks like. So if you guys noticed, it had a whole bunch of little wells in it, holes in it for the tubes, and it has a heated component on the top. So what's going to happen is we can change the temperature quickly in this machine. So it cycles, so thermo is heat, and it cycles the heat or the temperature. Okay, so here is a graph, and here's another little explanation of the steps in PCR. Okay, so first thing is we're going to denature. So here's temperature on the y-axis and time on the x-axis. Okay, so we're going to heat up to about 95. Okay, then we're going to cool down and we're going to let the primers stick to the target DNA. Okay, then we're going to extend, and that's when we're going to build out the DNA. We're going to build out this piece of DNA. So that is considered one cycle. Okay, so one cycle takes anywhere from 3 to 5 minutes or so. Usually we do about 35 cycles. Okay, so we'll do this 35 times. So 5 times 35, that's how long it's going to take to do run a PCR reaction. Okay, so here we're breaking down the steps again. So denaturation, so usually it starts with 1 to 5 minutes. Okay, it ensures that the double-stranded DNA is opened up. Okay, so the template DNA will come apart, right? And it'll be ready for the primers to bind to them. Okay, so then annealing, this is the trickiest one, okay, because you want the primers to stick to the template DNA, and you don't want them to stick to each other, and you, it's just like, it's a tricky step, okay, so the annealing temperature, okay, depends on the melting temperature of the primers, okay, and we're going to talk about that in a second. So primers are all a little bit different, okay? And designing primers takes practice. There are software programs that help you design primers based on what your sequence of DNA is. Okay, so during the annealing step, okay, the temperature of the reaction is going to decrease low enough that the primers will bind to the target DNA sequence. If the temperature is too low, they won't stick. If the temperature is too high, they won't stick. Okay, now, how do you find the temperature? Okay, so Tm is the melting temperature of the primers. Okay, so Tm is the temperature at which 1/2 of the DNA duplex will dissociate to become single-stranded, and it has to do with how stable these double-stranded DNA pieces are. Okay, it all depends on the sequence of the A's, T's, C's, and G's. Okay, so if you look at these base pairs, and you guys know C is going to pair with G and A is going to pair with T. Okay, when A pairs with T, there are 2 bonds. When C pairs with G, there are 3 bonds. Therefore, it takes more energy to break a C away from a G than it does to take an A away from a T. So it's harder, harder to break this bond than it is to break these bonds. Okay. Now, there are computer programs and they're free, and you just put in your sequence. You put in your DNA sequence that your primers are. Okay, so basically what you're going to do is you're going to take the information for the gene or the piece that you want to amplify. You're going to take the sequence and you're going to try to find what the information is on this side and this side of the gene of interest. Okay, then you can take the primers, you can put them in a database, and it will tell you, it'll calculate the melting temperature based on how many A's, T's, C's, and G's are in that sequence of DNA. So it'll tell you what temperature you should use to anneal. Okay, so when you go to make primers, you're looking for a piece of DNA where the C and the G content, okay, so the number of G's, guanines, and cytosines are in there. You want it to be about 40 to 60% G's and C's. If you have too many, the temperature is going to have to be really, really high to break it apart. Okay, so G and C here, okay, if you look at this and you put it into one of those programs, the G to C percentage is 43%, and the melting temperature is going to be 50. Okay, if you look at this one, it's 62%, so you got that many more G's and C's, so it's going to take more energy to melt it. Okay, and then this one is only 24%, so the temperature where it melts is lower. Okay, now, from the melting temperature, we can calculate what temperature we need to use for annealing. Okay, and there's this nice little formula here. So that the TA, the annealing temperature optimum, is 0.3 times the Tm of the primer plus 0.7x times the Tm of the product minus 25. Okay, so that's kind of crazy, right? They've made it simpler. Okay, so the magic thing is it's the melting temperature minus 5 degrees. Okay, so we want the primer to stick to the template DNA. We want it to be specific. If the temperature is too low, okay, it will stick accidentally to places where it's not supposed to stick. So you might get amplification of something that you're not supposed to amplify. Okay, so you want this optimum temperature. And if you're setting up a PCR reaction for the first time, you might have to play with this in order to figure it out. Okay, now primers, bad things can happen to primers when you design them. So sometimes primers could accidentally bind to each other, okay? So primers could bind to each other, and then they create what they call a primer dimer. Okay, another thing that could happen, and this, for some reason, this didn't get transferred over here, is primers can bind kind of like this, like a hairpin loop. So you just want to make sure that there's not too much homology between your primers. You don't want your primers to hook up with each other. And you guys will spend more time going over this if you ever have to design primers. And the programs that you use to design them catch and say, oh, look, this, you might have a primer dimer here, so don't use this one. This isn't a good choice. Okay, so here's standard PCR. So here's the original template DNA, right? And we melt it and we anneal. So the forward primers going one direction, backward primers going the other direction, and then we extend. Okay, so then there's the second cycle. So the second cycle, we'll start to get target DNA. We'll start to get these little pieces. And then by the third cycle, we're only getting the targets. Okay, so you don't have to memorize these numbers, but this is the idea. The copies are made exponentially. So they're made like two, they're made, so the first time you have 2, then you have 4, then you have 16. So after 30 cycles, you're gonna have whatever this is, how many, there's 9 zeros. So here's a million, right? And there's a billion, or I guess this is a billion, right? Now the things that can happen is that the enzyme can run out. Okay, the enzyme after 30 cycles does start to break down. So it's not going to go forever. You're going to run out of resources. But this is where you can potentially make a lot of DNA, a lot of copies of DNA. Okay, now problems. So here are problems. So with standard PCR and Taq polymerase doesn't proofread. Okay, so sometimes there are errors introduced, and if the errors are really early, so if the errors are happening out here, you could be copying something with an error in it. So that's not good. Okay, now other things that could happen is that if something is contaminated and the primers bind to it, they could cross-react. Okay, and you might be amplifying something that you don't want to amplify. So when they're doing PCR for the purposes of like genetic testing, you want to be really careful not to contaminate anything. Okay, now another thing, and you guys did this today, right, is with standard PCR, you have to run a gel and you're going to look for the bands after the reaction's complete. Okay, so you're going to have to look at it. So it's a yes or a no, right? Do you have a band? Or is there not a band? Okay, so it's not quantitative. Is it there or is it not there? Okay, and then this video link takes you to a video that just shows how PCR works and how the DNA is amplified. Okay, now PCR application. So there's a lot of different applications. It's used for everything right now. Okay, so it can be used for testing for genetic diseases. So if they know the gene that is defective, that's going to cause the disease, they can screen for it that way. Okay, they can also use it to test for infectious diseases. They could do it to see the relationship between you and another person. So are you the dad of the kid? Okay, we use it for probes if we're doing southern blots. We can use PCR to do sequencing, to study the relationship between different species. to look to see how genes are expressed and to figure out where genes actually are in the chromosomes. So I put this picture up here. This is COVID testing. So they take the test sample, they swab it, right? And then they take that DNA and they're going to try to amplify it. Okay, so there's an example. Now they're doing RT PCR because COVID-19 is an RNA virus. So we'll talk about that. Hang tight. It can be used for cloning. Okay, so there are cloning systems developed for PCR pieces, and that's called TA cloning. So the gene of interest is copied using PCR, and then you put it into a plasmid and transform with it. Okay, hybridization probes. So if you're looking for a piece of DNA, you can run a PCR and incorporate some sort of label into the DNA. So if you have A, T, C's, and D's, right? G's, okay, you could label one of these and you can actually see it. And I'll try to explain that more in lab. Okay, phylogenetic studies, this just shows relationships between species. Here's infectious disease. Okay, so why do we use it for infectious disease? So because culturing organisms, like if you have microbiology, it takes time and sometimes it's really hard to get things to grow. And some things can't grow without cells, right? So it's faster to be able to use PCR to look for specific sequences of DNA that are only found in certain organisms. Okay, so this says virtually all of the significant infectious agents of the human population have been identified. Okay, so they know the sequence. So if you know the sequence and you can amplify certain parts of it and say yes or no, whether somebody has it or not. There are some diseases that you can't use PCR because it's not an organism with DNA that's causing the disease. So for example, tetanus and botulism are actually caused by the toxins. So you can't screen for toxin DNA because toxins don't have DNA. So you have to look for the organism that might have caused it. Okay, now multiplex DNA or PCR is what we did in lab today. And I think you guys understand it. It makes sense with what we did. Some vocabulary that is in that lab that I didn't tell you before is amplicon. So an amplicon is the sequence that's being amplified. So that's the target sequence of DNA. Okay, so normally we only use a single primer set, but it's possible to use more than one set at a time, and that's called multiplex. Okay, so the important things are that the primers, you don't want them to bind to each other because that would be bad. They have to be specific for their targets and they need a similar melting temperature. Okay, so we did 3 sets of primers. So the 3 primer sets that we had, one of them was looking for the Cas9 target cut site in this LAC-Z gene. The other one was looking for where the DNA was inserted in the LAC-Z gene. So this would be a disrupted. And then the other one was just on a random place on the chromosome. Okay, so this was our control to make sure that when we did the InstaGene matrix thing, that we actually had DNA, right? So that you had DNA in there. This was the one to see if the CRISPR worked, right? And then this one was looking for an intact LAC-Z gene. Okay, so that's what we did. Okay, and I think that's it. I always get the extra page there. All right.